So yesterday we were talking about some mnemonics to remember the structure and function of all different types of antibodies. Today we'll go a little bit in detail. And remember, this chapter talks about an overview of antibodies, give you the basic concepts and basic properties. And obviously we will do that in detail as we move on in this series of lectures. So you can see again, another slide gives you some of the functions of antibodies. And remember that I gave you an example as well, that the body, the defense system needs to know who is coming in. Want to identify, want to react. And one of the reactions the antibodies help in is by those specific properties like precipitation, agglutination, immobilization, and neutralization. The whole idea is we want to save the damage. We have to protect the body or the organ. And be more specific, you can see over here, some of the antibodies may have all those function a little bit better than the other. Now, having said that, keep in mind that is coming from their half-life, that's coming from the structure they have, that is coming from their abilities that these antibodies have. And you can see some of the antibodies are really good in that. And as you remember, I said IgG is the senior most, abundant, about 80% in our system. And that basically takes care of almost all the neutralization that we have. So you can see we have different subgroups, subtypes of IgG, IgG 1, 2, 3, and 4. And then again, if you look at opsonization, IgG is there on the top, but IgA also participates. But some other antibodies may not have any function. When we talk of sensitization for killing by NK cell, remember, body has to respond. And if you remember me saying, the very first decision body has to make it whether it's self or non-self. So obviously, all bacteria, viruses, fungi, cancer, transplanted organ are considered as non-self. So body has to reject, or the other immunological term, body has to go for the big kill. That's what it does. And they are very specified cells that are going to do that. Antibodies basically assist in the process. So they are there, just like a mnemonic, I gave you an example. There's a family in the lymph node, which is like a home, is totally barricaded, very restrictive. It's not going to allow anything to come in. And the very first thing that if there is any foreign antigen or immunogen, the antibodies are going to react to it. Now, one of the things that these will do is that they can either tackle those intruders on their own, but remember, being very small in size, I gave you an example for those sisters, very fragile, and may not be able to do the complete job. So either they have to call their brothers, which are T cells, or they have to call, the uncles and aunts and dad and so on and so forth. So they cannot do it on their own, but they will definitely make the job easy for them. They can also call in complement, because complement, of course, is there to help. And then again, we were talking about some other abilities that you are interested in. For example, are they being transported across the epithelium? Because remember, since we are protected from exterior by skin and mucous membrane, so we wash our skins and we apply a lot of lotions and creams, so on and so forth. But there is an immune system in the skin that actually takes care of everything that touches our skin or wants to penetrate the skin. But on the other hand, our mucous membranes are vulnerable. We are eating, we're drinking, and they go through all that stresses. So there is something protective on the mucous membrane we call mucus, and that mucus contains antibodies. So there is an existing pattern. There is a part of innate immune response. It's already there. And if you remember, the most important antibody basically that is there on the mucous membrane is IgA. So IgA is there, very protective, that would not allow bacteria, viruses, fungi to pass into epithelium, because epithelium would be another route by this microorganism in giving entry into our system. So we are interested in transport across the epithelium. The other thing that we are interested in is that transport across placenta. Because remember, if mother is making an antibody, the question will come whether these antibodies are going to be passed to the baby in intrauterine life. So in this case, you can see IgG is the only antibody that is protective. There's a big sister that will go into the baby and be protective, and none of the others. Now the question you might ask, if we do, what they normally do is that in pregnancy, if they find something going wrong, so they would put a needle into amniotic cavity and take the fluid out and assess for presence of antibodies. Normally they expect IgG there, but if they find IgA, well, it will be unusual. Most likely they will look for IgM. If they find IgM in the baby's blood or amniotic fluid, that would suggest infection. So that's not good. Because remember, if as a rule of biology or immunology, babies are kept sterile in the womb of mother, highly protective. We don't want anything to go there. And the other question are diffusion into extravascular sites. Again, you will see that we have IgG, but IgA, and sometimes IgM can, depending upon infection. For example, you have rheumatoid arthritis, very common in this country. There's inflammation of the joints. So inflammation means injury. There's something going wrong in your joints. Now, immune system will assess that. An immune system needs to go and help you. That's the design of the immune system, because that is a defense system. So if it's going to go through that active site where this event of inflammation is taking place, apart from cells, they will deploy all their machinery of antibodies, T cells, so on and so forth. So in the process of help, so to speak, antibodies will also leak out of the lymphatic system, leak out of the circulatory system, and be accessible to those extravascular sites. And again, you can see, other than IgD, that doesn't want to go there, you pretty much will see almost all different types of antibodies there. And if you take the fluid out, that normally they do for diagnostic purposes, you will see the levels of antibodies there. And if you again find lots of IgM, that's why you say plus minus. If there is an IgM, that would suggest recent infection. Okay? And then again, we did talk about serum levels. You don't have to remember them, but just remember that some of them will be at higher concentration, because for a good reason. The other slide talks about isotypic, allotypic, and idiotypic differences among antibodies. Again, all antibodies have a heavy chain. So heavy chain is the one that gives the name. So gamma, delta, all these are heavy chains. And they have light chains. Light chains are variable. Right? And then again, remember I said there are 2 heavy chains and 2 light chains in every antibody. But having said that, the question that comes, what makes them specific if they're all the same? So keep in mind the distal fragment, which is like a fragment for antigen binding, Fab, is the one that will give that kind of complementary variation. So they are, I gave you an example the other day, like your fingertips. So they're all proteins, they're all sequence of amino acids. So pretty much sequence of amino acids in the heavier chain is the same. But the sequence of amino acids in Fabs differentiate. Okay? Now let's take an example. These are isotypic differences. When I say isotype of antibodies, what I mean is GAMDE. That's what I mean. They all are isotypes, but they are different. And there's a good reason, because they have a different heavy chain. So this one has a blue, just to give, they don't have a blue, but just to give you an idea, this is different than red, because they have a different heavy chain in terms of sequence of amino acid, in terms of genetics. Then you can also see that this is the variable portion on both sides. That's pretty much is considered as a white. So this means it can change. Now, differences resulting from the structural variation in heavy chain, that makes them, because if it is a gamma over here or micro over here, so that's a genetic makeup. So gamma, micro, delta, epsilon are the genetic, it is coming from the gene, that makes them different. Now, within the same one, like in IgG, you may have different types of IgG. So you have IgG 1, 2, 3, 4, at least 4 different types of IgG. So there are slight genetic differences you can see from here. Not a major, they still carry pretty much the same sequence of heavy chain. But there are slight differences between those IgG chains, as you can see depicted by the color, and that would give rise to IG1, IG2, IG3, IG4. So what's happening is that they are the same gene at a given locus. This pretty much distinguishes individual within a species, because if you all are human species, but you are different from each other. So that kind of differentiation between one species, because horses are one species, so you have antibodies from horses, cats, and cows, and so on and so forth. So within one human species, that kind of a difference that is there a little bit in your gene basically is called allotypic differences. And then finally, idiotypic differences. As you can see, they are different between the constant region due to expression of different heavy and light chains. So you have heavy and light chains. So you can see from here, the heavy and light chains are different. So you may have an IgG, which is from, for example, one species as compared to another. So what we normally do is, what in a treatment-wise, we, as I said yesterday, that we give an antigen to a horse, and the horse produces antibodies. We separate it on commercial basis, but the question will come, can we give it to human beings? The second question will come, will human beings recognize that antibody? So what sometimes we do it, that we keep the chain, like this one we'll keep over here, from horse or any other animal, but we'll change the fragment for antigen binding, but that's what your cells see. So we will, so to speak, call humanize horse antibodies. So we humanize horse antibody. That kind of stuff is coming from idiotypic differences that we normally see in commercial preparation of antibody. Now remember, so far we've talked about antigen, we talked about antibody. Afterwards, I'll take question afterwards. Well, all these tables will be posted tonight when I post you the audios, so everything will be there. Okay, immune reaction. So immune reaction basically means that an antigen wants to get into your system. And also remember, when I say that, I also mean food. I also will mean like in gastrointestinal tract, anything that you consume. By the stretch of mind, it also would mean in reproductive immunology sperm, because a sperm will be a foreign to a female because she has to accept. So everything is antigenic. Anything that has that computer tag is going to be considered something that our immune system is going to respond. But the most specific reaction that we have in immunology is called an antigen combines with an antibody and forms an antigen-antibody complex. So this is the reaction. It's called immune reaction. We already spent time on what an antigen is. Now we are talking about what an antibody is, and then once we do that, we'll talk about immune complexes. So that's where the problems come. I'll just give you an example. If, for example, an antigen enters in your system, antibodies want to take over or immobilize that. In the process, they clump, they aggregate, for example, in the blood or in the joints, and they settle somewhere. So the antigen-antibody complexes is something that cause problems. Sometimes one of the commonest reactions that you see is skin rash. So what's happening is you're taking a drug or food, anything as an antigen, antibodies react with that, they form complexes, and then you see those complexes sitting on your skin as all those red marks. And then antigen-antibody complexes will initiate inflammation as well, will initiate rheumatoid arthritis as well, will settle down and block vessels as well. So there are many things that are happening, but that's where it is coming from, and this is also the basis of the tests. So for example, once you come on Monday, you want to find out whether you are blood group A, B, AB, So what is happening is that if your cells have an antigen for A, we will give you an antibody for A, and they will react, and they'll form a complex, and we'll see under the microscope. That's the basis of the test that we have in terms of diagnostics. Now also remember, I think so far you may have formed this idea in your mind that there's an antigen, is recognized by an antibody, they kind of react and form a complex. But also keep in mind, the antibodies are looking at a sequence of amino acids on those antigens. And they are designed or made to go after them. They don't care what the antigen is. They don't care how large the antigen is, as long as that is expressed over here, right? Like this, this is a big table right now, right? So it's a part of inventory of the Chicago State University. But how would they find out? There's a tag here. So this is a very small piece of tag. That's where the scheme will come and tag it. It's going to take all the information there. So the antibodies carry that machinery to tag, right? So what may happen that they can also react to their own self. So you can make an antibody to another antibody because those antibodies are proteins and they also carry those sequence of amino acids. So sometime it may happen that an antibody within you is considered foreign by your cells. So they will initiate another cascade of reaction. You form antibody against antibody. Does that make sense? Okay, so this is an example. You can see there are two anti-idiotypic antibodies, AB1, and they are very specific. I mean, the cartoon they are giving is that they are very specific, like a lock, and you want to have a key. So an antibody should basically, like anti-antibody to this one, should snugly fit into that groove. Okay, and you can see by the design of it. Just an example, but actually it's a sequence of amino acids. So it's like DNA pairing that actually goes and sits over there exactly like interlock, like this one, and it's tight reaction. But also keep in mind that if there is another antibody, which is an antibody B, this was A, and this can only recognize a small portion of the sequence of amino acid expressed on that particular protein, so it will just bind there. But it would not solve the purpose, because the idea was we want to stop antigen-antibody complex so that we don't have a problem in terms of antigen-antibody complex mediated disease. So what's happening in this case, this was a good antibody because it did not allow an antigen to come over and get into an antibody complex activity. But this one, though it recognizes that, but it would still allow an antigen to come over and initiate the reaction. Does that make sense? So the idea is that you have to have a complete antibody, and you also have to have a profound antigen-antibody reaction to set the framework for immune response. And also keep in mind, immune response can be positive or negative as well. Doesn't mean that antigen-antibody complex will always cause harm. It will be beneficial as well. Okay, and next couple of slides, this is pretty much a reiteration, recap for what we discussed yesterday. I'm just gonna run through that. And this is based upon your understanding of that table, but I'll just point to you some of the things that probably you may wanna highlight that. So structural features of IgG is prominent in fluids. You can see from here, it has 2 gamma heavy chains. They are 50 K each. And most of the time, the light chains are kappa or gamma. Because right now we need to remember that the next lecture is more complicated, and that's basically a very important lecture, genetics of antibody formation. And this is one of those lectures that basically was, I think, year before last or last year, ACPE for accreditation people wanted us to teach all the pharmacists. It's all genetics. So we'll discuss that in detail, but right now keep in mind there are 2 important light chains, kappa or gamma. And then again, entire IgG is 150 kilodalton. The sediment of coefficient is 7s. These are something cramming material. But if you look at the structure, let's look at the structure of all. So just kind of visualize that, as I said about IgG, again, heavy chain, light chain, variable portion. And the other thing I would want you to notice in all the 5 is that I kind of made it in my chart like they all love sweets. They like sweets. So they need attachment of a carbohydrate moiety, glycoprotein. So there's like sticky ends. And they are important because many things will come and stick over there. They are proteins. But if you put glycon over there, glucose over there, so it is a glycoprotein. So you can see some of them have more. The bigger the size, I said M was fatty, heavy. So what it has is that it's basically composed of 5 different antibody molecules joined together by chain, which is called joining chain, J chain. So that's why I gave you a mnemonic. It's also called snowflake. It's chubby, heavier. But keep also in mind, if I was to ask you valency of an antibody. Valency is for the number of antigens that one antibody can possibly attach to. So let's put this as 1. If one fragment, which is a fragment for antigen binding, can hold on, like if I have 2 hands, if I can hold on to 2 different objects. So my valency will be 2. But if I was an IgM, so I have more hands. So technically, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This should be 10, technically, but that doesn't happen. The maximum it can take will be 5, and I'll explain that to you because of the size, it kind of gets restricted. It cannot move easily. If you look at IgA, again, pretty much the same. It has 2 molecules. It has a J chain, and it has got a secretory component. There's a secretory component which is very important for IgA, and it's easy for you to remember. I just told you that this antibody needs to be transported across the epithelium onto top of your mucous membrane as a part of your mucus. And if you lack that, then you are prone to infection. And then IgD and IgE have just a simple structure. Now, we already discussed about properties of IgE. I'm not going to go in detail. We already talked about that. And what I want to talk about today is that other than the half-life and so forth, that body has another mechanism, homeostatic mechanism of conservation. Highly conserved. It wants to save energy. So we need to, once we make IgG, like for example in figure 410, we want to make sure that we don't lose those antibodies. So the way we do it, so remember there are huge cells. So this is like one cell. This is a huge cell. And antibodies are good, like those little sisters. They are small, fragile. They go over there. They're small size, small half-life. They will grab two antigen, two foreign body. But what can they do? They cannot do anything. They can immobilize them. They can precipitate them. They can neutralize them, right? And what the other thing they will do is they will bring those intruders, antigen, to cells. They will bring it to the cells, right? And this is one of the cell. So this antibody has picked up two antigens. It will come down and stick to the cells. And remember, I said they will attach by Fc portion, which is a constant fragment. It is also called crystallizable fragment as well biochemically. Now, endocytosis. The principle for antibodies to get into the cell is endocytosis. Once they get endocytosed, then there is a specific protector receptor. This is an Fc portion, remember, this is a Fab portion, so there is a protector receptor, and I usually give an example of those young little sisters. They are tiny, small, so parents, whenever some intruder or a visitor comes in, they want to make sure that they're always there with the chaperone. So most of the activity you will see for this antibody is with chaperone, because they don't want to lose those antibodies. One such chaperone is specific over here to protect this receptor. It will attach over here, and then basically the idea is this will disentangle the antigens from the antibody. So antigens, like let's say two bacteria, will be separated. We wanted to deal with them separately and put on a lysosome, and the antibodies, we want to conserve them. And you can see that kind of dissociation takes place over here. The antibody again is carried by the protector and then taken away out to wherever it needs to go. So the whole idea is that they are very specific receptors, and you'll wonder, why do we need to know that? Because once we devise drugs for some of the actions, our drugs, our pharmacological agents are going to target them. So this is a pretty neat system in our body, that we have very specific receptors and very specific molecules that we have. And this basically is very important, because we used to wonder, why is it that only IgG can pass through the placenta and not any other antibody? The reason was, what we found out, that on placenta, there is only a receptor for attachment for IgG. And placental tissue, by the design of it, only carries a specific protective receptor for IgG. That is the reason that placenta can only pick up IgG and pass it on into the system, and not other antibodies. So that's a part of reproductive immunology. The rest of the properties of IgG we already discussed. One thing that I did not discuss, I just alluded to you a while ago, that antibodies are small, tiny. They cannot do all the job on their own. what they can do is they can bring that antigen or immunogen to cells. And that's why I give you an analogy. The cells are big brothers, uncles, and aunts, and so on and so forth, because they have this ability to deal with the intruder. So in this case, one such mechanism is called antibody-dependent cell-mediated cytotoxicity, ADCC. And this is basically a very important concept for you to understand if you want to understand allergy. So we'll just give you a rough idea today, and I have a full lecture on that. But the idea is that cells also need help from antibody. And then we talked about activation of complement anyway. Neutralization of toxin, I talked about that. Immobilization. So you can see all these antibodies have a lot of jobs over there. So for example, if this is a toxin or a venom, and it's like a molecule, and it has many different sites over here. So what will happen is these antibodies will come and attach to it on all sides. So one of the things that they would do will be immobilization. So this toxin cannot be absorbed into the blood. Or this toxin or venom, whatever, is going to stay at the place. So these are some of the functions of these antibodies. That's what we mean immobilization. For example, if you have one boil on your hand, and you know when we teach, boil is called by Staphylococcus aureus. So you have a tiny boil over there, but what is it that is preventing this boil to spread in your whole body? It can be systemic as well. So antibodies are there, just kind of rolling it up, holding it up over there, making sure that this particular bacteria is immobilized. It cannot get away from there. So these are the functions for the big sister IgG, which has a larger half-life as well. And we already discussed that earlier in the previous slide, but you can see from here, this specific receptor that I just mentioned is there. All those people doing research on reproductive immunology, that they find out that there is a time when during the trimester of pregnancy, there is an increase in IgG. Because remember, we need protective antibodies. The babies basically come with no information, with no immunity per se. The only immunity, they get it from the mother or the breast milk. These are the two sources of giving immunity, or vaccination. So if you take these three things out, then we'll have very high incidence of diseases. Because the whole idea is that we want to find out what is it that we can do to help, especially, for example, babies who are born premature. You just take a baby out of the womb of the mother, second trimester, third trimester. Does anybody know what was the smallest baby born? It's the size of the smallest preterm baby born. There was a big news, I would say, like 10 years ago. Many of you were not born then, right? 10, huh? Exactly this size, exactly this size. And you can Google it. She was born in Loyola University while I was there. This size of the smallest baby born and survived and still living today. So you can see the challenge that we have is that we need to find out what can we do to help, right? And also remember erythroblastosis fetalis that we discussed in physiology when we taught you blood. And again, I'm going to take that chapter when I teach you the lab as well. Why is it that if the mother is a rhesus negative, she can still give birth to the first baby from a rhesus positive father, but in subsequent pregnancy, she may abort it or kill the baby. So we'll discuss that. It's called erythroblastosis fetalis, where there is destruction. Now, remember that antibodies, because of the structures, would only go and attach when they have receptors. I already discussed that. So there is something called FC receptor. You can see receptor for the constant portion of the antibody. So there's a bacteria. Bacteria has all those antigens like thorns. So antibodies will go and block them. So, but they can't do anything else. Just kind of immobilize it, neutralize it. because then these thorns would not cause you damage, but then they will present that to phagocytic cells, and the phagocytic cells will obviously phagocytosis. So far we talked about phagocytosis, but we didn't talk about the detail as to what actually happens in phagocytosis. So this is the role of antibodies in phagocytosis. We talked about IgM as well. Pretty much we discussed everything, and one of the mnemonic I told you that was there, that they are very good at complement fixation. So they are very good at on complement fixation. And then again, they have, again, 2 light chains, 2 heavy chains, pentameric, because they have 5 different parts. Valency should be 10, but unfortunately, valency is 10. The other thing I want to talk about, and of course, take a lecture, is something we call class switching. So body is producing IgG, but you want body to produce IgM. So that kind of a message we want to do in vaccination and many different immunological processes. We call class switching. So that kind of class switching, where an antibody switches the class and changes the sequence of amino acid, this is one of the biological properties of IgM. Pretty much the same, we already discussed that. And the isoheme agglutinins present on your blood groups are IgMs. So once you come for the labs, so we will do that. So we're going to give you IgM antibodies, because they are IgM molecules that you can see from here. IgA, again, pretty much the same structure. We talked about that they are in all the secretions, size, sedimentation coefficient. We have 2 different types of IgA, IgA1 and IgA2. But 93% of the time on all our secretions we have IgA, and they are present, as I said, on all our mucosal surfaces. And again, very important for protection against invading microorganism. Now, the other question will be, and again, this is like a basic conceptual thing. You'll ask yourself, the antibodies are made in blood or lymph nodes, right? By cells. So the cells that make antibodies are B cells. Right? So that's what I said, the B cell was a mom, right? So we said a B cell. So B cell makes this antibody, but the antibodies basically are expressed and they locate on the top of mucous membrane. And that's like in our saliva. So what causes this transfer? So you can see from here, once they are made by the B cell, a B cell, when it wants to give birth to antibodies, it increases in size. It's called plasma cell. It is a B cell, but when we look under the microscope, it looks like larger. It's called plasma cell. So plasma cell give birth to dimeric IgA. Then we have a very specific receptor, it's called polyimmunoglobulin receptor. So this receptor is expressed on all those epithelial surfaces of respiratory tract, genital tract, and GI tract, nasal tracts. Wherever there's an epithelium, there is a receptor over there on the inner side of this epithelium. Again, chaperone. Remember the term I said, chaperone, because the antibodies are very delicate and they are there to be transported in and then expressed on the lumen with one special thing which is called secretory component on the top of IgA. And our bodies are producing it in tons at all times. And God forbid, if they do not, then we have a big problem. And we'll discuss that in a lecture. It's called immunodeficiency. There's going to be immunodeficiencies that we have. IgD, you can see pretty much same. They have same structure. They are co-expressed with IgM on the mature B cells. If you remember my mnemonic, and I said that IgM and IgDs are like small little kids, so they have to be carried by their mom and dad, so especially mom. So they only are stuck to their mom, so they're attached basically on the B cell. They have a lot of attachment. So if you look at B cell in our lab, when we look at B cell in our research lab upstairs, So what I do is that then I take two antibodies, one antibody against IgM and other antibody against IgD. So I will take these antibodies, put it on the cell, and they will get attached, and then I'll go and look at the flow cytometry and see if they are expressed. So that will show me that they are always there on the B cells. Okay? IgE, we talked about allergy. It is also important in parasitic infections as well. So allergy and parasitic infection, if you are prone to allergy, I'm going to go and ask for IgE. One other thing that's important, other than the structure of gamma and lepta chain, is that some of these antibodies, especially M and E, carry a additional domain, initial carbohydrate-like domain. So this is like attachment that you will see on IgE. Okay? Now, we know the antibodies are there, but antibodies follow a kinetics. Once you see a stranger for the first time, your reaction time is pretty much going to be a little bit longer because you need to identify, recognize, and respond. So once, for example, we give you the first shot of hepatitis. So your body has never seen hepatitis virus, hopefully. That's why they say that before they give you hepatitis B, they want to make sure that you have not been infected to hepatitis B before, hopefully. Because your body is going to see it for the first time. So first shot of hepatitis B, it will go into the system, body will recognize, antibodies will look at it, and then again, they will respond, but this will take processing time because you are not into the system. But of course, they will make some immune response. And the second time, when you give the same booster dose, now they already know that. So they are prepared to have an exponential kind of production of antibody. Okay? And then again, if you look at a graph for that, for example, I just give an example. This is your serum antibody concentration. That's what you normally do over time. And you look at your immune status right now. If I was to give you an immune shot of hepatitis B, I'll check your levels for antibody levels and want to make sure that you already don't have that because sometimes people don't remember. And then give you hepatitis shot. So there's going to be a latent period. It could be like a week or two because your immune system is taking that antigen, breaking it apart, presenting it to the antigen presenting cell. All these things is going on. You want to make sure that you properly identify that. And then they respond. The first response for any antigen is increase in IgM production. And you can see. But over time, IgM then declines. And then IgG kicks in. Because IgG is something which is a bigger sister, a bigger antibody, more half-life, a lot of function. We want to have an IgG in your system. But if I was to stop here and you say, I just took one shot, so your antibody levels are going to decline because your body sees that and it says, well, it is a threat, not a major threat. I'll make some antibodies. That should be good enough to take care for that. But body doesn't know that you want to have it for good for the rest of your life. So what you need to do is, so you have to stimulate, again, by a second booster. So you give a second immunogenic stimulus. And this should be at least a month or two apart. So you cannot have two injections at the same time. It will be of no use. You have to give body a time for recognition of antigen, process of antigen, and making sure it does respond. So if you give it for the second time, again, IgM will come in as an immediate response, and then again, your body will produce tons of IgG that should last for the rest of your life. So that's basically a principle of immune response. Does that make sense? Okay. Now, final slides. I talked about immunoglobulin. I told you it's a gamma globulin. So you may be wondering, maybe it's something to do with antibodies alone. No. we have a superfamily. Remember, I told you the example of the superfamily with mom and dad and kids and so on and so forth. So within such range in immunoglobulin, we have, because you need to know that there are chains there and there are proteins there. And immunoglobulin represent one type of immunoglobulins, but there are other members in the family, and they have characteristic kind of a loopy structure that we talked about, like globulin, and they have intra-chain disulfide bonds. So if you look at this picture, for example, and that, remember I said there are receptors on the cell, and we are going to go into real detail in this one. So if you look at, for example, immunoglobulin, and this is a bilipid cell membrane. I told you that the antibodies stick to B cells like M and D. So what they do is that they really poke their Fc portion right deep into the cell and stay tight. And this is what the structure is. But there are some other molecules, they pretty much are also Ig family because they have light and heavy chains and they have disulfide bonds to hold them together. So Ig alpha, Ig beta is a heterodimer. T cell receptor itself looks like an antibody because most of the drugs right now, they are in the market for cancer, for transplantation, for other, they basically are either targeting these receptors, either in terms of stimulation or blocking. So we need to know what is it that is present over there and we are blocking. So once we, in P3, P4 year, we're not going to go back and teach you immunology all over again. So this is your time to pretty much know that they are like this. The other molecules you can see also in the IgG family are CD3 molecules. CD3 molecules are expressed on T cells, and then you can see they have chains, gamma, delta, epsilon. And we talked about the genetic markers, which are called major histocompatibility marker, and there are two types of it, MSC class 1 and MSC class 2. And when you go for your tissue typing, when you want to donate an organ, they will run a sequence of this structure over here. They're going to give you the sequence of amino acids that you have, and the beta-2 microglobulin, and so on and so forth. And then there is something called polyimmunoglobulin receptors. So the idea is that the structures-wise, they look alike, but they have many things in common. So let's finally compare that the Fab portion of antibody is not the only portion that comes across an antigen. There are other things that have hands. So antibodies are not the only ones. Of course, important ones. So you can see antibodies, IgG, they have an antibody site. They look at the nature of antigen that may be bound, pretty much everything. And then they have affinity of antigen bind. That's another important thing, very important. I'll just touch upon that and let Dr. Doe talk about that in terms of affinity of antigen binding, because we want a drug to bind to that. Now, secondly, we have a T cell receptor. This will also attach to an antigen, and then again has an antigen binding site that pretty much have a sequence of amino acid, and then they are again different type of antigen binding. And then finally, the antigens will also be seen by MSC molecules, and they have also specificity, and they have also a affinity binding. But if you look at these three, you will see that some of them are very stable. So if I ask you in the question, one of my favorite questions is, I usually ask that if I was to present an antigen to 3 different antigen binding molecule, which one do you think is going to hold it tight? And what is MSC molecule? It's your genes. So any antigen that comes across your way, it really has to be seen by your genetic makeup before you respond. So that actually will be the basis of allergy once we talk about how to deal with that. Okay? So I'll finish here.